Good afternoon to everyone. Thank you very much for the invitation and for the organization of the Climed-Fruit of this seminar, of this project, which is definitely been very, very interesting. And we are, I think, learning quite a lot of things. And well, to me, it's quite hard to concentrate everything in 15 minutes, but I promise I'll try it because to talk about something that you've been working quite a lot since the last 10 years, it's quite hard, and I'm trying to sum up the main results of what we know about the grape leafhopper. I work as a researcher and development manager of this winery, of Bodegas Enguera, which is a winery located in Valencia, in the east coast of Spain, so Mediterranean climate as well. And the idea is to talk about the emerging pest in Mediterranean vineyards, particularly the grape leafhopper. I like to say that is kind of the new threat to Mediterranean viticulture because it's actually becoming a huge problem in some particular areas. Like I bet here it happens the same. The southern part of Italy probably is very affected by the leafhoppers. At least I'm in touch with some growers that report this problem, and as well as the southern part of France and many other places. So anyway, we get into the detail. The grape leafhopper is an insect. Through the whole seminar, we've been talking about different insects, orders, families. Some of them are very easy to distinguish outside, and in the case of leafhoppers, it's another completely a different story. It's very hard to identify the species of leafhoppers. And in this project, I'm going to show you how we do it and why we say Jacobias calibica, Ampoas cavitis, Asimetras caecens, and Ampoas caecipiens are the four species of leafhoppers we found in the vineyard. Family Cicadelidae, subfamily Toxicidae. So basically, what happened in order to show you what is the sort of damage and the behavior of these pests in the vineyard for the ones that you know the behavior of these pests, the biology. These insects, particularly in summertime, migrates to the vineyard, producing several generations of the pest, and within a few weeks, it can multiply by 10 their population and causing a huge damage in olives. Particularly at the end of August or the end of July, start to grow the population, and in August we saw this particular damage. So this is the numbers that we monitor, and this is a damage of leafhoppers in Turis, very close to Valencia. So basically, the insect sucks the internal tissues of the leaf, and the problem is that it can cause the defoliation of the vine. As you can see, not in this one. This vine has 80% approximately of surface affected by leafhopper. Later on, I'll let you know how we know that. And in this case, this is the drama. You can't imagine how hard for a vine is to ripe the grapes without leaves. So the quality of this grapes, is going to be very poor, and it compromises also the survival of the vine itself. So this, of course, happen only when the population are so high. But as I told you before, this is becoming an emerging pest, probably because of we've learned about this in the first presentation, which was very instructive in terms of the temperature and the different aspects of climate that affect also the development of this pest. Since the last couple of years, this pest is been developing in many different vineyards around Europe. So the objectives of the project was to identify the leafhoppers species in the vineyard, to understand the life cycle, to evaluate the damage in the different areas, and to describe the implemented strategies that the growers were doing to control this pest. This is if I have time, I'll talk about two different projects. This is the first one. And well, disseminate the results as many other projects that have public funding. Anyway, we are located in Spain, particularly in Comunitat Valenciana area. We went to 10 different vineyards, as you saw on the map. And we do, because it's very hard to identify the insects, we do a vacuum. We sample the insects and we do the DNA barcoding. And that was the strategy to exactly make sure that what we are identifying are the species that we determine. We set yellow sticky traps, is the way we monitor adults, and we also do sampling of leaves to monitor the nymphs. Apart from that, which is something we didn't see in any article, when you read about leafhoppers and how to evaluate the population, you see about the way adults behave, or you use yellow sticky traps to monitor adults, and you count also nymphs to understand the level of damage of the population. But very few articles are focused on the damage itself. So to identify, at least to sample the leaves and to scan them and to calculate the soil phase affected by leafhopper was crucial. So anyway, these are almond trees in blue, cover crops in black, rambla would be watercourse, and rosemarine, and the vine. So different places we went with a vacuum collector to monitor them or to know the species. As you can see in the vine, we find mainly Empoas cavitis and Jacobias calibica. And Jacobias calibica is also present in many other different crops or cover crops. It's interesting to see Asymmetrasca decedens because Asymmetrasca decedens is a species of leafhopper more related to almond trees. So it's interesting to see that not only in almond trees, but in the vineyard around almond trees are high levels of, or not high levels, because we cannot in this graph to talk about the abundance, but the presence, is what happened when you monitor with the DNA barcoding. Anyway, it's now easy to say that because in the past we didn't have the damage of leafhoppers, now the damage is not caused by Empoas cavitis, which is the most common insect species, but Jacobias calibica. So we say this damage is very uncommon, so it's caused by Jacobias calibica. But what we found is that we have Jacobias calibica, but we have also Empoas calibitis living in the vineyard. Normally, you can see the number of sequences we analyze. It's very low, and it's hard to extract strong conclusions from them, but at least it gives us an idea or a tendency that when we have a vineyard with Empoas cavitis, we don't have Jacobias calibica, and vice versa. It's also interesting to see this. Well, you cannot see this, but mainly the damage, and also there are some other aspects that influence the severity, the damage of leafhoppers, such as of course, the treatments, the conventional treatments, but the southern part of the Comunitat Valenciana, or the area, is the one that have the problem mainly of leafhoppers. You have to consider that this is data of 2023. In 2024, the damage is even higher, but anyway, this is what we have. And the main thing is it's a problem related to the south part or to the warm vineyard climate. Well, you cannot see anything, but I tell you that this, what we are comparing in these graphs is the conventional farming treatments. What happened is the gray leafhopper start to grow its population at the end of June, and it reached its peak in August. Once this happened, it reduces the population in September, but when we sprayed with chemical treatments such as acetamiprid or synthetic pyrethroids, these not only don't reduce the adults, of course, it kills them, but you see the population going up in a uncommon season, which would be late October, even November, to increase the population in a high levels. And the thing is that with the amount of treatments we are spraying in a vineyard like the one in the bottom part of the graph, which is Novelda in the south, we create resistance. So at the end of the day, it can reduce the spraying of chemical treatments can reduce the effect of the pest or the abundance of adults, but at the end of the day, it can also create an important amount of resistance. An interesting thing is in another project, which was the use of kaolin for particularly adapting the vineyard to climate change, we didn't see any effect on that aspect, on the adaptation of climate change, because it didn't have any effect on water potential, stomatal conductance, or even the production, and the quality of the so the bricks, pH, total acidity. We didn't see any difference because of the kaolin. What we saw is when we treat the vines, this was pre-verizon, so at the end of July, just before the grape leafhopper increases its levels, it reduced significantly the effect or the damage of the pest, as you can see in the graph with Tempranillo and Marcellan. We did the same trial in 2024, and we saw exactly the same thing. When we do one treatment, and that treatment was done only at the end of August, we didn't see significant differences between the one treatment kaolin and the control. And no matter if you treat with a dose of 5% or 10%, that the effect is exactly the same. It reduces between 30 and 35% the surface affected by the leafhopper. And talking about varieties, of course, it's we didn't discover anything new, but in the trial, because we had these four varieties, you can see that it affects significantly, or there are differences between the sensitivity of the variety to this pest. This was the experimental trial. This should have been in materials and methods, but anyway, just to show you the way we did the treatments. And although the sample is moderate because we didn't get a lot of sequences, we know that Empoas cavitis and Jacobus celebica are the most prevalent species in the vineyard, and leafhopper population develops exponentially at the end of June, reaching its peaks in mid-August, as I told you before. And it's a problem, an important problem in 7 out of 10 vineyards we monitor. And some of them, the growers didn't even know exactly what they had, because they thought, this was, humidity or something like that, So it's very unknown problem, and it definitely can affect, as you saw on some of the images, dramatically young vineyards. Of course, conventional insecticides can create and reduce its effectiveness, and the use of kaolin, just 5%, can reduce more than 30% the damage of leafhoppers. This was operative group of Comunitat Valenciana, that we work with these different wineries. We collaborate with different wineries and some research centers. And I would like to talk a little bit about this one, if I have some time, because this is very interesting. Apart from monitoring what we saw, okay, what can we do in an organic vineyard to increase the biological control of the pest? Probably I cannot show you the videos, but doesn't matter. We are basically we are basically introducing Chrysoperla carnea, a natural enemy, a predator, because, and why we introduce this predator and not the parasitoids, the mimarids, which are the insects that are related to the biological control of leafhoppers, basically because we cannot raise them, we cannot introduce them into the vineyard. But there are some general predators that in laboratory conditions, as you can see in this reference with Khalid Kif, that we work with them in this project. So he demonstrated that Chrysoperla carnea can predate leafhoppers. So the idea was to release them into the field and to see what happens. And what happens? Amazing. This is me doing the monitor of the news. Amazing. And this is Christina introducing the natural enemies. As you see, the video of the larves of Chrysoperla carnea. Okay. And this was the experimental trial. Forget about the right side of the image, which is Marsan and biodiversity. We are not focusing on that. We focus on Chardonnay control and Chardonnay with Chrysoperla carnea, because it's the same variety. That's the main thing. We cannot compare two different varieties because the sensitivity, although you don't see differences, there are differences. Okay, so we set yellow sticky trunks from May to July. We change them weekly. These 5 dots you see on the map. We sample shaking the shots, 50 shots per block, and we're introducing every 3 weeks 1,000 lace winds. So forget about the central bar, because the central bar is another variety. But if you compare Parcella chrysopas, which would be the introduction of lace winds, and Parcella control, which would be control of Chardonnay, you see differences in terms of the population. We see the yellow sticky traps, abundance. By the way, the levels are extremely high. 1,500 adults of leafhopper per trap per week is massive, the amount of insects we count. And in terms of the severity of damage, you can see the differences between the control and the second bar, which will be Parcella with chrysopas, lace winds. It's incredible. So to see this, help us to understand better the way this can help to increase the biological control. And you see here again the dynamic population of those leafhoppers in the 2 different blocks. And in the green side, the green line, you see the blocks where we introduce the lace winds and the moments we introduce them. And I'm going to talk about this. Well, actually, it's interesting to see that Jacobias calibica nymphs were the same in both places. So what happened when it comes to the dynamic population? When it comes to the dynamic population, you see that naturally what it happens is that Chrysoperla carnea has high levels in spring. It reduces a lot the levels of the larves in summer because of the high temperatures, and it goes down. It goes up again in I'm just finishing in September. But when we introduce them, we keep the levels of lace winds high, particularly in summer, where they are more ingredient to control or to increase the biological control of leafhoppers. So levels of leafhoppers are extremely high. Lace winds are major predators in the vineyard. Of course, the levels are very low in comparison with the amount of leafhoppers we have, and the introduction of natural enemies can reduce even 30% of the surface affected by leafhoppers. But you have to consider that this is a vineyard with very high level of leafhoppers. And thank you very much. to Generalitat Valenciana, which was, the local government that make this project possible. And thank you very much for your attention.